So we've covered the inside and also the layers of a prokaryotic cell. So now let's talk about the appendages. And again, just a quick review, this in purple is a single circular chromosome of this bacteria, which all prokaryotes have. This little loop is a plasmid, this rogue genetic element made of DNA. In blue you have the inclusions for storage. You have in red the ribosomes, which read the genetic instructions and build proteins from them. This is all floating in the cytoplasm. Yellow here you have the plasma membrane, or the phospholipid bilayer. Outside of that you have a peptidoglycan cell wall, because this is a bacteria. If this was an archaea, it would be made of a different substance. And then there's no outer membrane in this diagram, which means that this is a gram-positive bacteria. Remember, gram-positive bacteria have thick cell walls, gram-negative bacteria have thin cell walls plus an outer membrane. There's no outer membrane here, so this is a gram-positive bacteria. And then you have a capsule, which is made out of glycocalyx. Now there's all of these little appendages sticking out from the bacteria, so let's take a look at what those are about. We'll talk about flagella first. Flagella are whip-like tails that help bacteria move around. Flagella proteins are sensitive to environmental conditions and will move differently depending on whether environmental conditions are favorable or unfavorable for the bacteria. And this allows the bacteria to move towards favorable conditions and away from unfavorable conditions. Some environmental conditions that control or influence bacterial flagellar movement might be certain kinds of chemicals in the environment that represent nutrients or acids or something like that, oxygen levels, even light levels. So some bacteria have flagella that respond to light. And it just depends on the species of bacteria what kinds of environmental stimuli are going to change the direction of movement of that flagella. So we're mostly going to be talking about chemotaxis. So taxis, the term taxis, you can think of it like taxi. It means movement. So taxis are to move you around. So think of, to remember that taxis means movement, think of taxi. And then when the flagella, or when the bacterial movement is responsive to chemicals in the environment, we call that chemotaxis. So chemo for chemical, taxis for movement. But the other kinds of signals that we talked about, so if the bacteria is responsible to oxygen levels, that's called aerotaxis, or movement in response to oxygen, aero meaning air. Phototaxis is movement in response to light levels. We're really going to focus on chemotaxis in this class. Now flagella have a pretty complex morphology. We'll look at some diagrams here. So here is a gram-negative bacteria, and again you can tell it's a gram-negative bacteria because it has a cell membrane, just the normal plasma membrane that all cells have, and then it has an outer membrane here, so a second bilayer, a second lipid bilayer there. And here's the flagella, and flagella are anchored in both of these membranes for gram-negative bacteria with structures that resemble washers, nuts, and bolts. Here's a gram-positive bacteria. It's got a thicker cell wall, only a single lipid membrane, lipid bilayer. It's the same idea, it's just that it only has to be anchored into one membrane instead of two. There are other features of flagella that resemble machinery, including a rotating rotor, drive shafts, and a stator to stabilize the rotor. And you'll see this structure again when we look at ATP synthase in the electron transport chain, when we're talking about respiration and metabolism next week. So you can see, so here, this rotor, it spins. And it's the direction that it spins is controlled by, again, environmental signals like chemicals in the environment. So certain chemicals will cause the rotor to rotate one direction, other chemicals will cause it to rotate in the opposite direction, and that's going to control which direction the flagella spins. I provide links to these videos in the learning module so that you can see in more detail the mechanics of how flagella function. In this first link, which I won't include in the lecture, there's a little bacteria that you're looking at under a microscope on a slide, and its flagella is stuck on the bottom on the glass of the slide. And you can't really see the flagella, but what you can see is that the the bacteria spins because the flagella is anchored. So bacteria flagella don't undulate. There's an illusion of undulation because the flagella has this wavy design, but it's actually rigid. It's a rigid structure that rotates in a circle, and it does not actually undulate. So eukaryotic flagella, like the flagella on sperm, those do undulate. They're flexible. But bacterial flagella just spins. It's rigid and it spins. And so that's demonstrated in this, this poor little bacteria. It's got its flagella stuck in it. When it tries to move it, the bacteria itself is just spinning and spinning and spinning in circles. Oh, I'm sorry, that's this link down here. So I'll provide that link. This link at the top, it just shows you in more detail. It's just a video to music that shows all the components of the flagella fit together. All right, so taxis isn't just movement, it's directed movement. A directed movement towards favorable conditions and away from unfavorable conditions. Now, taxis does not require thought by the cell. It just requires a set of internal chemical cascades triggered by the external environment. So the way bacterial flagella work is when the presence of an attractant is high, and an attractant is something that attracts the bacteria, right? It's something that is good for the bacteria. So maybe like sugars, if this is a bacteria that digests sugars. So that would be an attractant, something that the bacteria wants to be around. So when the presence of an attractant is high, the flagella is going to spin in one direction, and that's going to cause the bacteria to run. A run is when the bacteria moves in a fast, straight line. So that happens when the flagella rotates in one direction. Now, a tumble is a random circling, which is how bacteria move when their flagella rotates in the opposite direction. So the way the undulating structure, the way that wavy structure of the flagella is put together, it's put together in such a way so that when the flagella rotates one direction, the bacteria moves in a straight line, called a run. When the flagella rotates in the opposite direction, the bacteria just randomly changes direction. It doesn't go in a straight line at all. It's just these random changes of direction. Okay. So bacterial taxis is perhaps best described not as intentional, but rather as a biased random walk. So we call this a biased random walk. Environmental signals cause the flagella to rotate one direction or another. And when an attractant increases, tumbling decreases, and the bacteria begin to run towards the attractant. When a repellent increases, something like an acid, a waste product, something that the bacteria doesn't want to be around, then tumbling increases, meaning that the bacteria start changing directions more and more. And they're changing directions randomly, but if they're moving towards a repellent, then any change of direction is going to be better than continuing in that direction, right? So when conditions, so to break this down, you can think of it this way. Favorable conditions are going to be when attractants are high and repellents are low. Any favorable condition is going to cause a run or a straight line movement. And the idea is if the bacteria has been moving in a good direction, it should keep going in that direction. If the environment becomes worse, if the bacteria starts moving into an unfavorable condition, and an unfavorable condition would be where the attractant is low in abundance and the repellent is high in abundance. So when those bacteria start moving into an unfavorable condition, the flagella is going to change direction that it rotates, and the bacteria is going to change direction. And it's just going to keep changing direction randomly in this random tumble until the conditions improve. It's going to randomly change direction until it just haphazardly moves into a better environment where attractants are high, repellents are low, and then it'll start moving in a straight line again. Okay? So this is all about just these internal chemical cascades that cause flagella to rotate clockwise or counterclockwise, and that results in the bacteria moving in a straight line when conditions are getting better, and to change direction when conditions are getting worse, and that results in the bacteria moving into better situations for itself. But again, it's not because of any kind of consciousness, it's not because of any kind of intention, it's just because of these chemical cascades. Now, this video I'm going to link to in your required videos. This is a really fantastic video of what we just described in that slide, but it has, it's dynamic, there's movement, and you can really see how the bacteria eventually get into a better condition by with flagella that work in this way. And I also want you to watch the 2 flagella movies, because I think that a lot of students coming into the class, they have this misconception that these flagella are undulating, and if you're not watching what I mean by them rotating stiffly, like it's a rigid structure that's rotating on an axis, it's not easy to understand this concept. So you really should watch some of these videos to really get a better sense of the mechanics of this. So the only thing I'll say about this video here, which will come up, the 2 flagella videos will come up next in your lecture requirements, video requirements. This video, I think the way it breaks it down is a little bit confusing, because it says, when the attractant is increasing, running is increasing, tumbling is decreasing. When attractant is decreasing, running is decreasing, tumbling is increasing. And then it does the same thing for repellents. The overarching theme is just when conditions are good, the bacteria keep going in the same direction, moving in a straight line. When conditions get worse, they start changing direction to change their situation. So now fimbriae are another type of appendage. They're like these little hairs that cover some bacteria. Not all bacteria have them. Not all bacteria have flagella. Not all bacteria have fimbriae, but some do. So fimbriae are like these little hairs that cover some bacteria, and I'd say they kind of look actually like the hairs that you find on burrs. Burrs, like those little seeds that have little spikes or hairs or stickers that make them stick to your clothes or to dog fur or something like that. That's what the fimbriae are like, those little hairs that make, because their most important function is attachment. So fimbriae help bacteria to attach to solid surfaces, or to attach to each other in biofilms, to attach to humans' host cells, to facilitate infection and invasion, those kinds of things. So the fimbriae, there's a lot of, when a bacteria has them, it has a lot of them. It covers their entire body. There might be hundreds to thousands per cell. These are much, much shorter than flagella and far less complex. They're just straight. They're not wavy or anything like that. So these are, like I said, they're used to attach to other cells or non-bacterial surfaces, and they often mediate first cell contact. So here's a picture of what fimbriae might look like. So you can see that this bacteria kind of looks like a burr because of all these, random little strands sticking out from it. Finally, there is something called a pilus. It can also be called a sex pilus. Now, prokaryotes, like bacteria and archaea, they do not engage in sexual reproduction, but the sex pilus is the closest thing to a sex organ that bacteria have. It can aid in bacterial movement, so it can play a role. It can act sort of like a flagella, or a little bit, or something, a clumsy flagella. But its main function is to enable one bacteria to transfer genetic material, a plasmid, to another bacteria. This is not officially sex, because in sexual reproduction, one full set of genetic information from each of 2 parents is combined. So when you have sexual reproduction, you have one full set of genetic information from one parent and another full set of genetic information from the other parent, and they're mixed together. Right? So the sex pilus only allows a subset of genetic information, just a tiny little plasmid, to be transferred from a donor to a receiving bacteria. So in this picture, you have 2 different bacteria. The one on the right has fimbriae, and the one on the left does not. So those are those little burr-like hair structures. And then you can see these 2 bacteria are connected with each other by a sex pilus. And you can't tell from looking at this which bacteria is the donor and which is the recipient, but one of these bacteria is transferring a plasmid to another. So they're not equally mixing.